So we're moving into our final talk of the day, and it will be by Caleb Allington. And the talk is Sample-Specific Contextualized Graphical Models Using Clinical and Molecular Data Reveal Transcriptional Network Heterogeneity Across 7,000 Tumors. Welcome.
All right. Yeah, all right. Thank you so much. It's been so fun hearing about these last two presentations where we've had a lot of genetic and other heterogeneity leading into different clinical outcomes for patients. So I'm going to continue that trend. So I'm going to talk about contextualized graphical models, which is our approach for inferring sample-specific graphical models that reveals transcriptional networks in 7,000 tumors. So first, why do we like the regulatory networks? Transcriptional regulatory networks are an interpretable representation of biological systems, explaining high-level phenotypes in terms of regulation and dysregulation. And so rather than having to model all of the molecular mechanisms and epigenetic factors that go into modulating gene expression, we can abstract this out into simple RNA to RNA interactions. And so this abstraction still retains things like the structure of a gene regulatory network and reveals things like signaling cascades and regulatory modules. So they're very useful for understanding the functional state of a cell or a group of cells. But inferring networks is generally hard without prior knowledge. We can do this for homogeneous networks. So for something like cells, which are all taken from the same cell line, we believe all these cells have the same transcriptional network. And so the gene expression samples that we get when we lyse these samples and perform RNA sequencing, we can use in some network learning algorithm to produce a network model with some guarantees about the underlying networks for the cell population because these samples are identically distributed. We believe that all of the regulatory networks that these samples were drawn from are similar or identical. Now, for heterogeneous populations, for something like a tumor-type cohort, where even in the smallest medical classification of a tumor, you will have broad genetic and epigenetic dysregulations leading to patient-specific mechanisms of tumorigenesis. And so for these transcriptional networks in these tumors, these networks are not going to look the same for any of our patients, and the gene expression samples that we get from these networks are not going to be identically distributed. So plugging these into a network learning algorithm, we're really not guaranteed anything about the output network or how well it relates to any of our patients. And so this asks kind of a broader question of how do we obtain or recover complex distributions when we only obtain one sample per underlying model? And so our answer to this is contextualization. So in comparison with traditional cohort modeling, where we have discrete partitions of our data, and we believe that this data was identically distributed in order to infer a model, usually by taking a statistic of this data that represents a parameter, contextualization views parameters as a function of context. And so by inferring this function, we don't have to have all of our parameters be identical, or the parameters be identically distributed. Our data can all be independently distributed. And by inferring this function, we can infer context-specific models. Or in the case where we have unique context for each sample, we can infer sample-specific models. So for something like learning a transcriptional regulatory model, an appropriate context might be genetic and epigenetic factors or proxy measures for these things. And so I'm going to give a quick how-to on contextualization with deep learning, because this is not just applicable to graphical models, but models in general. So first, define your differentiable objective of interest, or for a differential objective for your model of interest. So we'll just call this loss. Replace your model parameters with a differentiable context encoder. So usually for us, this looks like a neural network, which maps from the context space to the parameter space, where the outputs of this context encoder will be our context-specific parameters. And then an optional but useful step is to reparameterize the context encoder to reduce the model solution space. And so I'm going to throw this up, but we pick a smaller dimension k than the full parameter space and require that all of the context-specific models lie on this low-dimensional k-dimensional parameter space. And so this effectively reduces the solution space for all context-specific models, and it makes the learning problem a lot easier because it restricts the solution space. So all of this is differentiable, and you plug this into your favorite auto-differentiation library like PyTorch or JAX or TensorFlow and infer context-specific models. So for graphical models, the framework looks something like this. We have a patient context, which includes any number of complex modalities for that patient, including clinical information, imaging, genomic data. We plug this into the context encoder. The context encoder outputs to this low-dimensional subspace, which we'll call the subtype. This subtype selects which dimensions in this low-dimensional subspace to traverse in order to create this sample-specific model. These axes in the low-dimensional subspace are defined by model archetypes, or archetypal models that represent the axes in this smaller subspace the subtype is selecting from. And then we take a linear combination of these two to produce a sample-specific parameter set. We compare this parameter set using a network loss against the gene expression for this individual patient, and we get a loss that we can backpropagate through this entire framework. So we learn the sample-specific network, the archetypes, and the context encoder simultaneously and without prior knowledge. So we apply this to estimating transcriptional networks for 7,000 tumors. We use the Cancer Genome Atlas with 7,000 patients spanning 25 tumor types. We define losses for the correlation networks, Markov networks, and Bayesian networks, which we're going to infer with our contextualized approach. For context, we're going to use clinical information like patient demographics and the medical classification of a patient's tumor. We're going to have information about the tumor microenvironment with the biopsy composition, which tells us things like the percentage of normal and tumor cells in a sample, as well as the immune cell infiltration into that tissue. And we're also going to have some information about the genome and genetic information of that tumor with driver single nucleotide variations and somatic copy number alterations for that tumor. For the networks, we're going to learn these expression networks between 100 genes of interest from relevant cancer gene sets. And then for the Bayesian networks, which are a little dimensionality limited, we're going to pick a hallmark transcriptome dysregulation metric, which is the mean absolute deviation from average healthy expression levels for 25 cancer hallmark gene sets. Okay, and so we qualitatively evaluate this approach first by embedding all of the individual modalities we used in this approach, as well as the graphical models using UMAP for individual patients. And we're going to color these patient embeddings by their tumor medical classification and see how well the medical class of this tumor is represented in the topology of the data. So first, the clinical, the contexts that we use, none of these views identify or separate the medical classifications of the tumors very well. As expected, the transcriptomic view gives a pretty good separation of the tumor type, with the hallmark dysregulation muddying this quite a bit. And for the network views, we find that this also separates the medical classifications of tumors very well and also produces subclusters of these tumors. Of these tumor types, notably in the thyroid carcinoma, the uterine endometrial carcinoma, and in brain lower grade glioma. So here we have highlighted the brain lower grade glioma, which splits into 3 subclusters across the network subtypes, or across the network model types, where the subtypes of the cancers are not apparent in any of the other modalities used in this approach. So digging into this a little bit more, we replot for just the brain lower grade glioma patients. And so we can see that, one, the network subtypes are highly consistent across the network model classes that we have contextualized and inferred for every patient in this data set. And two, that the network subtypes are not separable in any of the other modalities here. So we perform a hierarchical clustering on the network model, specifically the Markov networks in this figure, and we find that these overlap with a well-known clinical subtyping of brain lower grade glioma. So this cluster of clusters subtyping includes information like histology, the specific down to the nucleotide position mutation in these genes, enrichments in other gene mutations, the microRNA data, clinical information, and there's one more I'm forgetting. But anyway, we use none of the microRNA data. We don't use histopathology information. And we also have a binarized indicator of mutation where we don't have this specific co-deletion that's highly predictive of the subtypes that they have for this cluster of clusters approach. And so we find that, one, our subtypes are organized by the regulatory module organization in these Markov networks, and two, we get this broad overlap with this clinical subtype. But it's not a perfect overlap. And so we're kind of interested in seeing why are these patients reassigned from the cluster of clusters subtype and the network subtype. And so we find that our network subtypes, when compared with the previous state-of-the-art subtypes, produce an improved survival split for brain lower grade glioma. So the most prognostic subtype, or the subtype with the worst prognosis, we remove these outlier patients when we view this disease in terms of the networks, and so the survival split is much more drastic. And so this has been a nice qualitative evaluation of the approach, but we'd also like to do a quantitative evaluation because these, at the end of the day, are statistical models that we can evaluate in terms of error with baseline approaches. So we're going to have 3 baselines that we evaluate this against. One is the population baseline, where we take all of our data, our heterogeneous data, and plug it into one type of model, or one model. We are going to have a context-clustered baseline, where we perform an unsupervised method of personalizing our data, where we're going to cluster based on the context and learn an individual model for each of these clusters. We are then going to take a supervised approach to personalization, where we're going to split based on the medical disease classification, which is, we believe, the most informative feature for each of for the models. And then we're going to compare all of these with our contextualized approach. And so we find that across all of the network classes and across all of the baselines, contextualization improves model accuracy. But again, the motivation of this approach was to show that we can do either sample-specific modeling or modeling of rare diseases or smaller subpopulations that are underrepresented in our training set. So we also compare this for undersampled or rare types of diseases. So in comparison with a larger disease set, the brain lower grade glioma, which we have 373 patients for, we see a reduction in the error of 30%. We see similar reductions in error on the smaller sets of patients for uveal melanoma and B-cell lymphoma. Notably, for the B-cell lymphoma, we are only fitting a model to 36 patients to learn these networks. And so the small sample size for the disease-clustered models, which are only given these 36 samples for training, drastically overfit to the training set, whereas the contextualization prevents overfitting on these network models. And so networks are a rich and interpretable method of representing complex systems, revealing modularity, functional organization, and opportunities for intervention. And contextualized networks enable network-based analysis at sample-specific resolution. So some things we're excited about with this approach are, first, modeling beyond sampling constraints. So inferring networks that are transcriptional networks that represent the state of individual cells. With the data set recently published, the SNARE-seq data set, we have paired ATAC-seq and RNA-seq data. And so we can use this ATAC-seq data as a context to infer the gene regulatory networks for single cells. Also, we would like to discover biomarkers relating to differential regulation. So genetic and epigenetic biomarkers are difficult to create outside of notable exceptions like the HER2 biomarker for breast cancer. And so we'd like to use these networks to identify what expression biomarker, or what expression levels might be qualified as biomarkers for specific regulatory states in tumors or other heterogeneous diseases. We also have this context encoder, which estimates the effect of genetic and epigenetics on network spaces. So these networks are a simplified model that represents the RNA-RNA interactions at an abstract level, but by quantifying how these RNA-RNA interactions change with genetic and epigenetic changes, we can infer what the effect might be on the overall regulatory system. And finally, this framework generates these networks from context. So we would like to predict regulatory changes under potential treatments. So by including treatments or time in the context for these networks, you can imagine that for an unseen treatment combination, we can infer what a regulatory network might look like under this unseen combination. Similarly, if we are inferring networks over a course of treatment or over time, we can generalize to future time points or roll back the clock to infer what the cancer regulatory network looked like in the presence of either fewer mutations or different microenvironments. And so finally, all of this was done with the Contextualized Heterogeneous Modeling Toolbox, which I developed. You can pip install Contextualized-ML. You can visit us at contextualized.ml. And we have a nice sklearn style wrapper for these where you can import, fit, and predict in 4 lines of code to infer a population of context-specific models. We have this implemented for Markov networks, correlation, and Bayesian networks you saw today, as well as linear models and classification models. So if you're working with biological data and you're using modeling approaches, odds are there's heterogeneity in your data set, and you can use Contextualized to try and explain some of that heterogeneity in a model-based way. So this was a collaboration with a broad number of institutions, but I'm very happy to acknowledge my collaborators, Ben, Tom, Jackie, Manolis, and Eric, as well as the researchers and developers who have made Contextualized possible, Wesley, Joanny, Alyssa, and Daniel. And yeah, with that, please visit us at contextualized.ml and check us out. Thank you.
Great, thank you. Questions?
Thank you. That's an awesome talk. It's finally great to see a graphical model showing up here. But so I'm going to ask a question. So this contextualization, is it something that's similar to like local graphical models where like with specific input values to your networks or like graphical model, it could potentially changes the structure of the networks or like the dependency structures in the IMAP or something like that that's in the model? Or is this integrated into your like say Markov random field or your Haida Markov model? Or is this like a separate model that you have to learn, like this contextualization?
Yeah, so the focus is shifted from learning individual models to learning this meta model that describes the relationship between the parameters in models, but we're still satisfying we're optimizing for a statistic, like an objective function that relates to the probability of these models. So, I have a slide that shows, these are the objective functions that we're actually maximizing here, where they broadly relate to these Pearson's correlation, Markov networks, and Bayesian networks. And so you could use these models the same way you would use a model inferred by plugging all of your data into any of these individual optimizers.
So basically you incorporated this contextualization into the likelihood function already.
Yes.
For these.
Yes, exactly.
That's awesome. Thanks.
Yeah.
Any other questions?
Yeah, awesome talks, thanks. My question is, like, for the network archetypes, are they like predefined or they are learnable parameters?
Yeah, so the question is, The network archetypes are something that's really exciting about this because, one, they're not predefined. They're learned through the same backpropagation workflow that we learn with the context encoder. And two, they actually do represent meaningful biology. You have a low-dimensional space that allows you to traverse the full population of context-specific models. And so there's been some work on interpreting these archetypal parameter sets for linear models, but for the network models, we're less clear on that. But that is an active research direction because representing the entire model space in low dimensions seems like it would have some implicit meaning, and there would be some new biology you could find there.
Okay, I think we can take one more question. Yeah.
It's really exciting that you are integrating context with the genomic data sets, but with like a variety of different cancers. Is there a universal set of features that go into the context, or is there some pruning that happens?
Yeah, so we do this for a data set where we have all of the information we use for all of the cancer patients. So that includes the somatic copy number variants, the mutation calls, the tumor composition and immune infiltration, as well as the medical classification and clinical information about those patients. So we have this for all 7,000 patients, and we use it broadly to contextualize these models, but because this framework is, we have a set input to that neural network that this is learned for, and it's optimized for that set of inputs. It would be difficult to add or subtract different types of data when you add or subtract a different tumor or a different tumor type into your data set. So if we had trained this with all of the information that we did, and then all of a sudden we have a new cohort of patients that have histopathology images that we'd like to include, it's difficult to do that.
Okay, great. Thank you. One more round of applause for all the speakers. Thank you.
Great question. Thank you.